Okay, let's do this. So what I've done is I've clicked through down at the bottom of the article. There's a thing that says supplementary information, and you click it, and then it takes you to another page that says you want to download the supplementary information. Here's all the stuff that's in there. So I've known people who work at the same place, not this guy, but one of the others that he works with quite frequently, who say that when they are reviewing a paper, they generally don't really pay much attention to what's in the paper itself. They are only going to be looking at the supplementary information, which is all the extra stuff, all the technical detail that you couldn't fit into the paper, or that maybe isn't considered like important enough to go into the paper, but is totally important enough that it has to be there for people that want to be able to read it. And so they usually make these as like an online supplement. You can click through a thing and you can get to the supplement. Now that's an extreme view, only reading the supplementary material, but it's also a good judge of quality because you get to see all the things that somebody's maybe trying to hide or, that didn't work out quite as well. And you get the details. And the devil is there, but so are all the angels. So let's go have a look. And so you'll get a page like this, and I would say include this in what you're considering. You're welcome to anyway. So this is from the Scripps Research Institute, which is where I used to work, one of the places I used to work. All right, and so they're going through all the methods up here. Useful if you need to know how they're doing the neutralization assay. For example, here is the neutralization assay. Bam. Okay, what are fancy things? Let's see. Tissue culture infectious doses. There we go. So this is actually using live virus, and they're using a TCID50 is you take your virus, and this is a way to count them. You put in virus and you say, okay, that infected the cells. You put in less virus, less virus, less virus. If the virus infects the cells will end up just destroyed after 1 to 2 days. About 48 hours is pretty good for total cell destruction of all the cells that are there. And so you just take these plates with little tiny wells in them, and you just do lots of different dilutions. So you have it normal, you have it diluted, cut in half, and a quarter, and an eighth, and a 16th, so on, until you reach a point finally where you no longer are infecting the cells. And then the point at which you have infected 50% of the cells, or you have a 50% chance of infecting the cells, and you calculate this mathematically based on the data that you get. That's what they're doing. So using 100 TCID50s, so enough virus to start roughly 100 infections. And they are going to try to neutralize these. Now, some of the other papers that we use are only trying to neutralize 50 TCID50s. And so this is worth considering. So the numbers we get here will be like more conservative or maybe more strict. We'll say they'll find out they need probably about twice as much antibody as you would need if you were doing a 50 TCID50 test. Yeah. So, great. And they're going through exactly how they're doing it and how they're looking at it. ELISA, this is how they're going to read out whether or not they have successfully bound the antibody to the spike. And so it would be worth you looking up what an ELISA is. This is something that we are going to cover. And it's pretty basic, but at the same time, if you haven't gone through it much before, it's going to seem new. It's not that hard, though, thankfully. So I'll leave that for you on your own to dig up. Okay, so down in here, let's look at all the supplementary figures on this real quickly to give you a nice idea. So the antibody that this paper is about is called CR3022, and I don't know what that stands for, but it's some laboratory designation. They probably tested a lot of things. They may have tested 3,022 different antibodies, and you know what? This one worked, so great for them. What they're showing here is that so you can actually look at the antibody, and you can go back to the genome of the cells that made that antibody, and you can say, okay, which variable region was used, which diversity region, which joining region was used, and which heavy chain was used, or which, constant region was used, VDJC. You can figure out which one was used. That's great. But then the other thing that B cells do is they will take the basic sequence and they will put in random mutations. And if the random mutations make it better, then they'll keep going, and they'll keep putting in more mutations until it doesn't make it any better. They'll divide, and so there'll be lots of different versions of the B cell with a kind of a weak hit that are modifying, just randomly mutating to try and get a better hit. And then whoever comes up with the best hit, that's the one that's going to go out and form the antibody. So this is the original sequence. The antibody here has this particular sequence, and so it's actually got 1, 2, 3, 4, 5 changes in that part, and 6, 7, 8, and so on in this other part. So that's what they are showing. And they're showing the region that was mutated and what the mutations were. Okay, next thing, and this is absolutely key to this paper. So this is what they call the epitope. An epitope is whatever an antibody binds, as far as this paper is concerned. Yeah, that's a good enough definition. So it's like the antigen, but particularly the exact configuration of parts on the antigen that are bound by this particular antibody. And so you can see the thing that we're binding is all of these. And what you've got here are a lot of F's and Y's. And F's and Y's are both aromatic amino acids, which means they've got some of these electrons running around these what they call aromatic rings in chemistry. And these electrons are not going to be localized. So sometimes they'll be double bonded here, sometimes it'll act as if it's double bonded here. They'll just be in constant state of flux. They can do additional kinds of binding here that you don't really get with other antibodies. And so this is the thing that the antibody is actually recognizing. Now the numbers on the bottom, so K means lysine and 378 means lysine at position 378 of the spike protein. So that's neat. Y 380, but look at these things. I don't think you will find 2 amino acids. Nope, there's 2. I don't think you'll find 3 though that are actually adjacent to each other. So the things the antibody is binding are actually coming together from different parts of the protein. And the rest of the protein has to be there, or at least the rest of the receptor binding domain, in order for these things to end up in this configuration, which is exactly what the antibody is recognizing. Now, shown down here, this is the actual antibody. This is the part that's going to recognize those things. And we've also got quite a few of these hydrophobic, not just hydrophobic, but aromatic. There we go. Aromatic amino acids. We got a tyrosine there. We got a tryptophan here. And we got another tryptophan over here. Yeah. And we've got, looks like, oh yeah, another tyrosine over here. Great. And another tyrosine over here. So we've got a bunch of aromatic hydrocarbon side chains that are binding to a bunch of other aromatic hydrocarbon side chains. These are all going to be pretty hydrophobic. And so this is a face that's going to be mostly driven by hydrophobic interactions, but also some what they call hydrogen bond interactions. And so that's actually just showing them, and here they're showing you some of the parts. So epitope is the part on the spike protein that the antibody is going to bind. Paratope is the part of the, like the face of the antibody that actually binds to the spike. So this is the spike, this is the antibody, this is the paratope, this is the spike, this is the epitope. There we go. Okay. So what you got here is the result of, I believe, something called surface plasmon resonance. And what we're doing is trying to see, can we get interaction with between the spike receptor binding domain and the antibody. And if we do get interaction, we should have a dose-dependent increase. And so this is some binding right there. But if we put in more concentration, we should get more binding, more binding, more binding, like that. And so this is showing that, yeah, they are binding, and this is pretty normal, and they're calculating a dissociation constant. So what does it take for this thing to fall off, which tells you kind of in a backwards way how well does this bind. You can see for SARS coronavirus 2, you've got a big spread, and so you do get more binding as you put more in. For SARS coronavirus 1, you've got a really tight spread up here, which means that the antibodies don't have to cooperate. Each one just latches on kind of indestructibly. So this is what really tight binding looks like. This is what kind of loosey-goosey, but still good binding looks like. And yeah, so we got a KD at 115 here for this loose binding, and a KD at about 1 nanomolar down here. This would be equivalent of tight binding. So basically the lower the number, the tighter the binding with these particular stats. All right, so let's look at these things. So neutralization activity. So here's the actual results of the neutralization test. They show a little bit over there, but this is the full test. And so they're going from not diluted to diluted 3-fold, to diluted 6-fold, 12-fold, and so on. And you can see doesn't block, sorry. Totally blocks, what, about 25%, and doesn't block. So our 50% blocking point will be somewhere in here. It'd be around a dilution of 10 micrograms per mil, which is pretty darn poor. So this is going to be like a negative control, this M396. Here is, let's see, S230, and you can see this one is... So, okay, this is actually pretty good binding. So, fewer micrograms per mil binding and neutralizing 100% means good antibody. There we go. Okay, so this is a reasonable antibody. This is a great antibody because our 50% neutralization point is somewhere below 3.125, somewhere, there-ish. CR3022, you can see up here, neutralizes SARS coronavirus 1 reasonably. The 50% point is somewhere between 50 and 100 micrograms per mil. So you need, compared to this one, you might need like 1. So it's probably 50 times as weak as this one. And it's probably about, I don't know, where's our point? Like 10, probably about 5 times as weak as M396. There we go. But it does neutralize to some extent. And you can see these things all neutralize SARS coronavirus original, but not SARS coronavirus 2, even though this thing does bind. So it neutralizes on regular SARS coronavirus, binds and neutralizes, binds and does not neutralize on SARS coronavirus 2. Because it's weaker binding and because there's a difference. There's a glycosylation site that SARS coronavirus has and SARS coronavirus 2 is missing. And think of that glycosylation as like a little bush. It's actually part of what the antibody binds, and it's bending that thing down flat, and it's making a different, tighter, bigger binding site that probably it's the glycan, the actual sugar, that is blocking ACE2 from binding. And when that sugar's not there, the antibody still sticks, but it's no longer able to just barely block ACE2 from binding. So there we go. Here is, let's see, here's the whole spike protein. Here's the open conformation of the receptor binding domain. Here is the receptor binding domain at the top, and they're showing you where the epitope is. So it's not at the top, the bit that actually binds to ACE2, which is right there. It's kind of on the side of this thing. And yeah, it is able to bind the up conformation in SARS coronavirus 2. And let's see what else we've got. Here we go. So last thing, this is, let's see, CR3022. And so they're showing it. They only did the experiment with this antibody and the receptor binding domain. But they're then taking this thing and sticking it back onto the entire spike and saying, okay, if the rest of the spike was here, what would happen? And with regular SARS coronavirus, you would get a clash. If the antibody's bound, then the antibody would be running into this thing down here, which would limit the movement of the N-terminal domain and limit the unlocking so that this little receptor binding domain, which is the pink part, can go up. So the N-terminal domain is in brown, the pink part is the receptor binding domain, and the antibody is just going to nestle right in there and bind to the N-terminal domain and block the receptor binding domain, they think. Of course, they've only got data for this little part, but they can model it in because they know what the rest of the protein looks like because we have the structure of that from other groups. and they're saying they use the this particular model for measuring the conformation. Okay, last little thing we'll cover. Every paper that has a X-ray crystal structure will have a table like this. This is the most important table for understanding the X-ray crystallography and whether it was done well. Important parts to pick out. So these things at the top are just characteristics of the place where they went to do the experiment. You want to know that it's a good place and it had a good enough X-ray beam. So fine. The space group is going to be different on every crystal. That's just the way that they pack together. Let's see. Unit cell parameters, that's how big each repeating unit of the crystal is. And so they're pretty big unit cells, actually. And then the resolution here. This is going to be a key. So it's going to go from a low resolution of like 50 angstroms, which is, you know, a protein just looks like a blob at 50 angstroms. An angstrom is about the size of one atom. They're going to resolve all the way down to 3.1, and the last shell, where the last bit of data is coming from, is going to cover from 3.2 to 3.1. And so you take that and say, well, okay, that's compared to other crystal structures of proteins, this is terrible. But compared to antibodies bound to protein crystal structures, this is right in the middle of the pack. This is pretty good. Antibodies are very difficult. It's very difficult to solve the structure of an antibody on a protein of any kind. And this lab happens to be really good at doing it, but normally you could get well below 1.5 angstroms is about, you know, like normal, workman-like. Anything below about 2 angstroms is considered pretty reasonable. So this is okay. To show you here, I've got another tab that's open here, and it actually shows you if we let it load. There we go. So what this means is the amount of uncertainty in the structure. So a 3 angstrom structure, like we've got here, the blue sort of hashed edge, this is all you can see in the structure. And so from this, you try to fit the protein inside of it. And you can see there are bits that just don't even fit well. Like this entire bond and this carbon are left kind of outside the blob that you can see. But as you improve the resolution, you can get much more precision. And you can actually rotate this a little bit, and it'll still fit inside this giant cloud, of possibility of where it could be. And same thing over here. So pretty terrible. And as we decrease the resolution, or increase the resolution, go to a smaller resolution, like the best structures, about 0.5 angstroms, this is like an ideal, would be, you could see just little circles where every atom is. So you're basically looking at the electron clouds, and you're even smaller than the distance between the bond. Yeah, you can't even see the bond, which is pretty cool. Somewhere around 2 angstroms, you can get a pretty good fit. Anything under 2, and yeah, look at that. You can tell this is a tryptophan because it's got the 2 holes in it that I'm pointing to here, and the 2 rings, and this is the only amino acid that does that. So you can actually see individual amino acids in your structure below 2, and at 3, it's a kind of a, You take it because you have to, not because you want to. But some people will try to push and get a lower angstrom resolution by using kind of crummy data at the edge, at the borderline. And so in that case, so they're saying it's about 100% complete overall. That's the 100 here. But how complete was the data in the last shell at 3.2 to 3.1? Well, in this case, it was 100% complete. So they are being very conservative. They could have gone out and grabbed some trashy data from the next resolution shell outside of this, and they could have got a lower nominal, named resolution. Better. We say higher resolution for a smaller number. So they could have got a higher resolution, smaller number, by including junk data, but they didn't do that. So they're being conservative here, and that's fine. I'm okay with that. And so, let's see. The other important bits here are going to be, what's the resolution? So 50 down to 3.1, fine, we got that elsewhere. Did they do a lot? Did they count a lot of reflections? So this is a good number. You want this to be high. And the other parameters in here are things that you'd have to read up on protein crystallography for them to make a lot of sense. They're measures of quality, though. And so this is pretty good. And so this entire assembly, this entire brick of proteins that they've got together, has 4,900 atoms in it, of which 4,900 are actually the atoms, and 30 are ligands, which are things that are sticking to the atoms that help them make the crystal originally. All right, and so table 2, which I think might be, well, we're getting close to the end. We're actually looking in the crystal structure, and we're saying, okay, what is bonded to what, and how close are these things? And what kind of bond is it? Is it a hydrogen bond? A salt bridge is between a positive and a negative charge amino acid. And so these are pretty reasonable distances for those bonds. If they were trying to say that something that's 5 angstroms apart is totally hydrogen bonded, and you knew much about physics, you would say, no, that doesn't work. Hydrogen bonds at that kind of length are super weak and probably not meaningful. Let's see. And you know what? Let's cut her off there. This is... yeah, this is something that you don't need. Okay, so there we go. That's the stuff, the data behind the paper. And now we're going to go look at the actual paper. Papers in science are written in this very compact style. They try to minimize the amount of wasted space. It's almost like a telephone directory in the old days. They are just cramming as much information onto a page, high as data density as possible. They try not to have any words that they don't need in there. And so these are sometimes a little bit hard to read, but they're a real good way to build up your understanding by looking up all those terms that they use. So there we go. That's our paper intro for the week.